he took it.
Then after they broke up, we liked them better than her, so we...
Okay, I guess we'll get started. Thank you, Hardys, for coming back for a third session. This one on cell replication, and it has an attractive and more or less accurate picture of it. An open section of a strand of DNA with about 10 base pairs between each full twist. So these next two seminars are going to deal with the two most fundamental functions of a cell. They're referred to by biologists as replication and transcription-translation. Replication occurs when a cell makes a duplicate copy of its genome while dividing into two cells. Transcription-translation involves retrieving information from the cell's DNA and using it to assemble the complex molecules the cell depends upon. The detailed understanding of these processes began in the 50s and required uncovering and understanding the role of deoxyribonucleic acid, DNA. While the names Crick and Watson are well known to, presumably well known to you, a great many scientists played major roles in bringing about the marvelous detailed description we have of these processes. And we're still learning more. We'll start with replication because it's simpler. And to get underway, we'll need to get better acquainted with DNA. DNA, I'll say it again, deoxyribonucleic acid, is essential to most forms of life on Earth, be it, bacterial, plant, or animal. It's very large, in a sense it's going to be a very long molecule, encodes the blueprint for the formation and maintenance of the entire creature that carries it. DNA is a stable, inheritable blueprint that transmits the identity of the cell, but flexible enough to allow for evolutionary change. Clearly, DNA has to be capable of storing a great deal of information, and it has to store it in a secure and compact format, as it has to fit into each and every cell. The genomic information is encoded in a sequence, and that idea of the sequence is critical, of 4 different chemical substances called nucleotides. Those nucleotides are adenine, thymine, guanine, and cytosine. Of absolutely critical importance is adenine and thymine only bond with each other, as is the case for guanine and cytosine. That's true within the DNA. As shown below, DNA has a double helical structure where the nuclides where those nuclides bridge between the two structures. Where am I here? They bridge across the two strands in that fashion. DNA folds up, allowing it to fit into the cell, as you saw in those watercolors of good cells last week, where the DNA was all folded up so it was able to fit into the middle of the very tiny cell. The DNA of an E. coli bacteria, which is going to be the subject of our next two lectures, is a bacteria, and it has 4,639,221 nucleotide connections that make up for the prescription for 4,401 genes, and holding the direction for the assembly of 116 RNA molecules and 4,285 protein molecules. Those prescriptions are encoded in particular sequences of the 4 nucleotides and direct the assembling of the large molecules essential to the cell's functioning and reproduction. So here's a look now at what DNA looks like. Here you'll notice they often give a form to adenine and thymine. This semicircular form here shows that this will, whoops, will mate with thymine, and this triangular shaped thing shows that cytosine and guanine will go together. Looked at in somewhat more detail, you see there is going to be a sugar phosphate backbone to these to the DNA that make up this spiral structure here, and here are the 2 nucleotides, thymine and adenine, and their bonding is a weak bonding. It's just simply a hydrogen bond between those two molecules. It's a double bond. It's a double bond here, double hydrogen bond. Whereas in the case of guanine and cytosine, the bond is made by 3 hydrogen bonds. So the guanine cytosine bonding is a little tighter than the thymine adenine. You might wonder, why does this thing twist? And as far as I can see, the chemistry of that is not fully elucidated. The words that are used is that the nucleotides are hydrophobic, and so they want to get as far away from water as they can. So you can imagine they would curl up inside this double strand as it tightens. So you can imagine, as you played with string yourself, that it's really a very delicate question as to how much tension you put on the string, as to whether it just simply neatly strands or starts to bunch up on itself. So the whole problem of the structure of this DNA and some of its complexity is, I think, a thing that's still being worked on, even though it's so basic. So in something like a bacteria, this DNA is just one big, you can think of it as a circular piece. Of course, it can't occur as a circle in the cell because it's just too long, so it has to be folded up in many ways. But it is a single unbroken strand of these nucleotides, and as I said, there was something like 4,600,000 of these base pairs that make up the genome of an E. coli. Our genome is the order of almost 1,000 times bigger than that. It's something like 3.6 billion base pairs, but whereas something the order of 90% of the DNA in E. coli prescribes for the making of particular proteins that the E. coli needs. In our case, the information to encode for we have about 20,000 proteins in our body, but it's only 1 or 2% of that total genome. So there's a great deal of the human genome that, while we have written it down and recorded it, we don't truly know exactly what its function is. Let's see. And just happened to mention that there's a particular E. coli strand, K12, that's used as kind of a base reference for examining various E. coli versions, and it goes back to a strain collected at Stanford in 1925. So how is this genomic data written down? Well, you can imagine if you've got something like 4.6 million pieces of information, it's not awfully useful to make a table of them because referencing them would be extremely delicate. But that's basically the way they're known, and of course, as you might imagine, all this stuff is stored in computers and is far more readily accessible in computers. In this case here, I've written the genome associated with 3 different kinds of E. coli, and their position, the C stands for cytosine, A for adenine, G for guanine, and T for thymine. So just the whole thing is written down. And it's just amazing how they figure out what these things are. It's nothing like I would have imagined as a physicist, that somehow you have a string of DNA, you read it in, and you have a thing that tells you exactly which one of those things is the next in the sequence. It's a very, very complicated job of just chopping up the DNA and putting it back together in ways that strike me as absolutely ingenious and requiring, as well as great intelligence, great patience. Looking some more at the structure of this stuff, here again is the helix. These base pairs are a bit sparse on this. There is an attendant ribonucleic acid called RNA, and the deoxy means without oxygen, and that's because in RNA you have this on, oh my goodness, you have in this formation of the sugars, I guess it is, there's an OH branch over here, and in DNA that OH is simply hydrogen, and I'm given to understand that this is ends up making for a more stable kind of a bond, but it's chemistry beyond anything I really understand. So this location of the carbon nuclides in this strand, looked over here, you start here, and this is given position 1, 2. Carbon is understood to be at a place where nothing is written, 3, 4, and 5. So that turns out to be relatively important, because anytime you look at DNA, you see that it's set up in such a way that one end of it, or one strand of it, is indicated as 5' and over on the other side it's 3'. And the reason for that is the way the bonding goes. This is the location of, oh boy, this is the 5 location here on the strand, and it goes down to bonding with the 3 location over on that side. And when you're on the other way, on the other side, it's 3 to 5. So in a sense, you can think of this as running 5 to 3 this way. Oh boy. And this one runs in the opposite sense, 5 to 3 going this way. So they're oppositely oriented strands. One running this way, 5 to 3, and this one runs 5 to 3 in this direction. Turns out to be important because some molecules that we'll encounter can only read in one direction. They can only read in the 5 to 3 point, the 5' to 3' direction. Questions? Henry?
What's the significance? Oh, I don't really need that, but what are the bonds between guanine and cytosines stronger than the bond between the adenine and the thymine?
It's stronger because there are 3 hydrogen bonds.
What is that of significance? What's the significance of that?
I'm not entirely sure, but it does turn out, for example, that at the origin of the E. coli's DNA, most of the stuff that's there is adenine thymine, and it's weaker and hence easier to open in the region of the origin when the replication begins.
So it's nothing to do with the twisting.
No, it might. They seem happy enough to just simply feel that saying that these nucleotides are hydrophobic and hence end up hiding is a reason you would twist the thing to close them off from they're in a water environment. In a cell, if you just have DNA out dry, it doesn't necessarily fold at all. The folding is a consequence of it being in solution.
Yeah, a whole bunch of things. There was a lady who did, I think, X-ray crystallography on which Cricket Watson depends, and I think she really got cheated out of a Nobel.
Yes.
Do you recall who she is?
Rosalind.
Okay.
Franklin.
Help me.
Rosalind Franklin? Is that her name?
Franklin.
Yeah.
Rosalind Franklin?
Yeah.
Thank you.
Okay. And then you said the RNA is less stable than the DNA.
Yes.
Which makes me speculate that maybe that's where some of the mutations come from. That it gets messed up and translates the wrong information on by.
I don't think I want to go there yet.
Well, I'm not asking you if you know that. I'm just speculating.
But the RNA doesn't really have a job in basically doing replication. The replication, the RNA simply is encountered in the oh boy.
Kind of reconstruction.
Yeah, in reconstructing proteins. It doesn't have anything to do with the issue of replication.
Sorry to dominate all this, but
No?
a human has way more of these proteins and everything than a human has way more proteins and so on.
Yeah.
And we ascribe a whole lot of our behavior to genes.
Yes.
So I assume that some of that is building genes that give us a lot more behavioral flexibility than your one-cell critter.
Well, it turns out, interestingly enough, that the size of the genome and the complexity of the creature are not correlated. I mean, there's a very rough correlation, but I think some of the very largest genomes are held by some Japanese water lily. And it's about 10 times larger than our genome.
Wow.
Here we go.
I just wanted to comment on
Please.
the question of RNA stability. I think it's a really important point that's worth holding off on. But some genomes, like those of some viruses, are made out of RNA. And they do have the stability problem that you raised. So it's important to keep that in mind, and something that I'm sure Gerry will come back to.
Yeah. Okay. Now the fact that the bonds between the nucleotides are hydrogen bonds and therefore weak, they're the order of 10 or 20 times weaker than covalent bonds. For example, a covalent bond is the order of 7 electron volts, while the hydrogen bond is only the order of 0.3 electron volts. But however, the whole DNA molecule is extremely stable because there are millions of those weak linkages in it. So you can imagine that it's really actually pretty tightly put together. However, while the whole thing is kind of stable, it can be attacked locally with the appropriate chemicals and opened up at that point, and the information that's contained inside it can be exposed. As you can see, DNA is a chemical, and we know a great deal about it. The complete genome of thousands of hundreds of thousands of bacteria and their variants are known. In 2003, the first complete human genome was completed, and there have been more than 1 million since. But just recently has the complete genome of a single person been recorded. Remember that, or recall that the human genome consists of 3 billion base pairs. So while we understand the role of the DNA in an E. coli, we only clearly understand the role of a much smaller part of the human DNA. But we'll talk more about that later. So you can see the detail in which a genome can be known. They're specified by a particular nucleotide at a specific location on the genome. The rate and volume of genetic data that is created by biologists now is so vast and so rapid that in order to digest it, a whole new field called bioinformatics had to be created. Bioinformatics stores the information and advances ways that you can make it accessible and digestible to the researchers in the field. As DNA is a molecule, it should be able to be produced in the lab, and indeed it has. The current state of the art can reliably produce pieces of DNA that are about now 5,000 base pairs long. However, they have to be pierced, however they can be pieced together using various CRISPR techniques. And in May of 2019, some British scientists produced an E. coli whose entire genome was artificially manufactured. The note accompanying the paper said, "The bacteria are alive, though unusually shaped and reproducing slowly. But their cells operate according to a new set of biological rules, producing familiar proteins with a reconstructed genetic code." That was an article in Nature in 2019. So startling as it may be that you can create this whole DNA, you have to remember that's not the creation of life. Life requires a cell. You have to take that DNA and put it piece by piece, that artificial DNA, and put it piece by piece into a living cell in order to find out that you had successfully fabricated. So, of course, for any of the information encoded in the cell's DNA to be useful, it has to be, you have to get it to be accessible. And we'll see two instances of how that information is accessed and used: replication of the cell and assembling the cell's proteins. Replication is the simplest, so that's what we're going to start with. Cell division is perhaps the most fundamental process in biology. Most cells are produced by cellular division, with some differences between eukaryotes and prokaryotes. The primary objective in cell division, obviously, is the preservation and duplication of the original cell genome. Before cell division can occur, the genomic information that is stored in the chromosome, DNA, has to be replicated. And that duplicated genome must be clearly partitioned between parent and the progeny. A great deal of cellular infrastructure is involved in ensuring consistency of genomic information among generations. We will show how one bacteria becomes two bacteria in a process termed binary fission. So the cell's bacteria reproductive cycle can be broken into three periods: B, C, and D. Why they chose B, C, and D, I have no idea. There is a letter A, maybe they thought it was too commonly used or something like that. But at any rate, B is sort of the period where a cell gets enough nourishment and gets together the pieces that it needs to do the job for replication, and it's rather variable in time. It makes a lot of difference whether there's plenty of nutrients available to the bacteria. Now, however, on reaching that sufficient level of growth, the cell's ready to replicate. And at that point, a standard process takes over. That little figure below shows you about how the process goes. So the first thing that it does is it ends up making a copy of itself. And here is shown into what was just one complete DNA. Now there are two complete copies in here, and the cell proceeds to undergo now the physical separation in order to create the two cells. The period to me was really surprising that in many cases the maximal period of the cell reproducing itself is spent in copying its own genome. It takes about 20 to 30 minutes for an E. coli to copy its genome. It takes a human the order of a day to end up copying that 1,000-time bigger genome. And the length of that genome becomes, in some cases, a rather crippling factor. For a salamander in Georgia has a great problem in that it is almost always born with physical deformities simply because it ends up breaking up, the cells break up before it's completely ended up copying the entire genome. It's a really messy and horrible situation. So how does that go? So the origin of so you have to go and find the origin now of this big piece of DNA in an E. coli. Remember, it's something like 4.6 million base pairs long. And at its origin, it has a specific identification. There are 245 base pairs. They're long, and they're rich, as I had mentioned before, in adenine-thymine base pairs. And having fewer base pairs, those things are a little weaker than the guanine-cytosine sequence. And so when the cell has accumulated enough energy in the form of ATP, remember that is that triphosphate that is the energy source, a typical energy source in a cell. And it gets together a density of a thing known as DNA-A. When that reaches a sufficient level, the DNA gathers at the origin of that big DNA genome and starts to assemble the molecular complexes that will execute the replication. The bacterial DNA replication begins by relaxing the coiled up chromosome with a thing called, well, there you go,
Topoisomerase.
Henry.
Topoisomerase?
Thank you. An enzyme called helicase, I can handle that, before separating the DNA strands by breaking the hydrogen bonds between those base pairs. So here's this, whatever it is, getting the thing a bit unwound, and then the helicase goes to work, and it's able to undo or dissolve those hydrogen bonds between the pairs, and you start to open up that DNA, and you're going in two directions. So imagine you've got this big circular thing, you've now opened it up, and you're starting to pull it apart, going around, and you're going to eventually come together at the end, and the job will be completed. But before you get there, the goal of replication is obvious, is to make an exact copy of the cell genome. And it's made doable by the fact that the thymine only bonds with adenine and cytosine only bonds with guanine. So having opened up the DNA at the origin, that a DNA polymerase that you haven't seen yet constructs a new strand matched with the existing template strand by virtue of that unique bonding that's I've just referred to. So wherever it finds an adenine on the strand, it picks a thymine from the cell and throws it into the strand. The bonding is affected by that triphosphate coming in, making the bond as shown here, and dropping off a diphosphate, which is then recirculated and used in making more triphosphates. Now, what's important is on one strand So on the 5 strand, things can go readily because that DNA polymerase puts things together only in a 5 to 3 direction. So on the other leg, which is 3', it has to it opens it up, and it can't do the bonding any further. So what happens is pieces, here I am trying to remember it all, ends up creating elements that are about 10 nucleotides long, and they're oriented in such a way, 5 to 3, that the primase, the DNA primase, can bind them to the existing strand. So what's happening as the thing is opening up, the original single strand is being made into 2 identical copies by the polymerase on the DNA, fishing out pieces that look very much like this, that have the sugar bond and one of the bases that you need, the correct nucleotide to match. So it is set up so that it's fishing out a cytosine to go and match with guanine, and hence making an identical copy of what was a single strand now is getting to be is becoming a double strand that is identical with the initial strand. Sorry, but it's not all that complicated. I'm sorry.
Understood.
Oh, yes, it is.
So, and as I said, it takes the E. coli about 40 minutes to carry out that copying of those. So it's clipping along. Each one of these is clipping along at the rate of about 1,000 per second. But remember, there's one over on the other side, so the overall rate is actually twice that fast. So it takes about, as I said, about 40 minutes to make that copy. And having completed making the copy of the DNA, the cell now proceeds to grow larger. So here we show the cell sitting there rather dormantly, getting healthy enough to the point that it says, okay, I think I can become 2 cells right now. So it ends up copying itself. And here is the other strand now that's been copied and separated, and then begins a process where the cell can physically separate and become 2 copies of what originally was a single cell. And of course, it's absolutely critical that when you've gotten to this phase, that you stop doing any further reproduction. You have to really wait for this thing to get ready again, to get strong enough and with enough nourishment and energy to be able to undergo the process once again. So if you remember back, let's go back and take a look at what von Neumann said. How is it satisfying the requirements that he set. Well, a robot must carry the complete blueprint for its own construction and maintenance. Apparently, it does have that. It has something that reads the instruction. That is, something knows, I better get ahead, find that origin, and start opening things up, and then the DNA polymerase or the replicon ends up creating the duplicate of the genome, and then the robot, of course, has to have a second copy of that of the blueprint. So we end up now with 2 blueprints. Now, so what to me was surprising is this process of replication seemed to be treated in a relatively straightforward way, like it just seemed like, oh, you put the you've got the replicator or whatever name you chose to give the thing that was going to do the replicating, and have it do its work. But my gosh, that thing that it's dealing with is the cell's DNA, and it can't go to sleep for 20 minutes while it's end up replicating. So there's action going on the DNA all the time, mostly in the form of RNA opening up the DNA and creating messenger RNA that would be used to further make the proteins that the cell needs to stay alive and carry out any other functions that it needed. E. coli seems to have found a very simple way of dealing with those things. As it's speeding along, when it runs into one of these RNA polymerases on the DNA, it just knocks it off. It just bangs them off and continues on with the replication. That knocking off that RNA every now and then makes a break in the cell that can make a break in the strand that has to end up being repaired, but the cell seems to know how to do it. So here are just what I was talking about. Here is the RNAP. In one case, it's running into the replicator, bang, head-on collision. In the other case, it's headed in the same direction. But in either case, E. coli response, and it's not necessarily typical, is to just simply knock the RNA polymer off the DNA. Now, the whole issue there of replication is tied into the issue of what's the error rate. And high fidelity of that replication is not just merely important, it's essential if you're going to preserve genetic identity and to prevent just the accumulation of harmful mutations. So, but nothing's perfect, and so errors are unavoidable. In something as simple actually as an E. coli, there are various proofreading procedures that end up correcting the errors that are made in the primary, just going ahead, that splitting open and fishing pairs out and hooking them in to the thing. The error rate is somewhere between 1 in 10,000 and 1 in 100,000. There's a first stage of repair that fixes that up by about a factor of 100, and then just checking for what should I say, regularity in the DNA reduces the error rate by another factor of the order of 1,000. So the DNA replication fidelity is good to something like a part in 10 billion, which is quite remarkable. But you shouldn't believe that that doesn't allow for mutation, because an E. coli has 4.6 million base pairs, and if the error rate is 10 to the minus 10, and the bacteria replicates every 40 minutes, then in the course of a day, there's about a 1% probability that there'll be a mutation in that bacteria. So within any community of bacteria, there's just bound to be many, many mutations. Here's an example of the kind of thing that happens. As you know, the typical way that the base pairing happens is typically thymine couples with adenine and guanine, and cytosine with guanine, but apparently there are a few cases in which those connections get made, where thymine connects with guanine, cytosine with adenine. Those particular malconnections are pretty much covered up in the initial stage of proofreading. So while the process is, fortunately for us, quite accurate with high fidelity, there is enough variation that even in the case of non-sexual reproduction, variation is possible. And as I said, next week I'll show you the ingenious way that a cell manages to assemble the molecular parts that it needs to sustain itself. So, questions?
Sure.
Donna?
Well, we have good bacteria. We have good bacteria and bad bacteria. Bad bacteria, okay?
Yes.
And I always thought that E. coli was a bad bacteria.
Well,
So I'm wondering if all of the different bacteria reproduce themselves according to the same plan, or if E. coli was a larger cell, maybe
so
bacteria.
you have a lot of E. coli in your gut.
Yeah, right.
And the E. coli in your gut is absolutely essential. It's good stuff. It's when you take it in some other form, like accidentally, you might end up getting some orally, or breathe it in, and it would end up in other than your gut. And then it can start making trouble. So the good and bad bacteria, Lowell.
I think it depends on the strain of E. coli.
Uh-huh.
That's what determines whether they're pathogenic or not. I don't think it is your method of ingestion. It's whether or not you have a strain that's pathogenic to humans. There's thousands of strains of E. coli.
You bet.
And so the question of pathogenicity relates to what strain it is.
Uh-huh.
If you take a harmless E. coli and you ingest it and you drink it, it's not going to hurt anything. It's in your gut.
Uh-huh. Okay.
But the other thing I wanted to to point out from a simplistic point of view, when you look at that process of replication, you end up with a new two-stranded DNA. And it's important to realize that one of those strands is from the original DNA molecule, and the other is the newly formed half of that molecule. So that you begin, the two strands separate, and then there's replication, so that in each of the new DNA molecules, there's one strand of old and one strand of new. I don't know if that makes any sense or not. But that's the simplest way of looking at that process of replication.
Try it on me again.
It's called conservative replication. Or it used to be, when I did this 50 years ago. Semi-conservative, sorry. Do you want to answer for him?
Yeah, I think the point's well taken that any DNA molecule historically has one strand that was a parent,
Yes.
and the other strand that was the daughter strand, as it's called. So there's an old and a new. And this is one of the beautiful conceptual frameworks that physicists, Max Delbruck, gave to this process. He was able to build the three possible models of all the three universe of models in these three concepts, and one of them turned out to be the correct one, semi-conservative. I do want to make a comment on two of these points. What changes a harmless E. coli into a pathogenic one is often information from another bacterium that comes in as a tiny chromosome, a plasmid. So this kind of so-called horizontal gene transfer is going on in bacteria as well as higher organisms, and that's how it can become a dangerous one wherever it lands in the body.
Okay. Thank you. Now,
A question on
there's a microphone coming.
a question on bioinformatics. It's my understanding that somebody's tinkering with using DNA as a storage device or mechanism for computer use. Does anybody know anything about that?
I've heard about it, but I don't know anything about it. It certainly would be powerful to have, something that has four values rather than two.
And a huge amount of storage in a small space.
Yes. That's certainly true. Yeah, I mean, to think that, let's see, our DNA is something like 3 billion base pairs, and in each of us there are 30 to 50 trillion cells. So yeah, it's a very economical way to store data.
Yes, we're done.